Welcome to the Ashby Village Science and Ideas Group. I'm Joe Evinger, and I help coordinate this group's events with the help of our co-host and tech expert, Hillary Naylor. Sarah Evinger also helps us with the recording. All members here have varied educational and work experience backgrounds, but we all share a common interest in science and new ideas. We currently have over 70 members in our group, and another 100 or so on our guest list. We meet via Zoom the second Thursday of every month from about 3 o'clock until 4:30. We have a different guest speaker each month who speaks on a topic related to science and ideas. Today we have Denis Titov. He will present on a talk entitled Adventures in Metabolism and Nutrition. I want to tell you that in the coming months, next month, we have Alan Bruno, who will speak to us about managing California's water resources. In May, we'll have a talk on how we domesticated fire and fire domesticated us. And towards the fall or winter, we have a speaker who will talk to us about his new book, Most Delicious Poison, about poisons. We're always looking for new speakers. If any of you personally know someone, please recommend to them to us along with their contact information. Hillary will give you my email too. We invite all Ashby Village members to join our group. Others interested in Ashby Village or just simply our presentations are also welcome. Ashby Village is a 501c3 tax-exempt organization. I asked Hillary, please, to place a link to the Ashby Village website in the chat. To our guests, please consider a donation to Ashby Village or joining our organization via the website. Now, Sarah Evinger will now introduce today's speaker, Denis Titov.
Hi, everybody. Denis is an assistant professor at the University of California, Berkeley, with joint appointments in the Department of Nutritional Sciences and Toxicology, the Department of Molecular and Cell Biology, and the Center for Computational Biology. His lab is interested in understanding how changes in metabolism affect aging and age-associated diseases. Denis received his undergraduate degree in chemistry from Novosibirsk State University, Russia, and his PhD in pharmacology from Johns Hopkins University, where he investigated the mechanism action of bioactive natural products. He then completed his postdoc fellowship at Massachusetts General Hospital, where he pioneered novel approaches for studying and manipulating energy metabolism in living cells. Dr. Titov's long-term research goal is to understand how metabolic homeostasis is achieved and how caloric restriction extends lifespan. How does diet extend lifespan and delay the onset of age-associated diseases? Can we predict the diet that will increase the lifespan of an individual animal? Titov's lab is using a combination of biochemistry, mathematical modeling, physiology, custom instrumentation, and novel tool development to study metabolism and aging in mammalian cell culture and model organisms. To you, Dr. Titov.
Okay, thank you. Thank you for the introduction, and thanks for inviting me here to speak. Very excited to tell you a little bit about the work that we do in our lab. So like Hillary said, please ask questions anytime. The more this is a conversation versus just me speaking, the more fun I think it will be. And also I apologize in advance. I typically talk to just metabolism scientists. And so there will surely be a bunch of words that I use that I think everybody knows the meaning of. That will not be true. But this is where, stop me anytime and ask questions. And so what I thought I would do is I would just give you kind of two examples of questions that my lab has recently kind of partially answered to give you an idea of what we do and maybe how we do it. And so the first question is, it's a sort of a century-old puzzle about why different cells that grow very fast exhibit something called the Warburg Effect. And I will tell you what this is and what the puzzle is about. And the second question is, how does caloric restriction, which basically means eating less calories, extends lifespan of this interesting organism called C. elegans, which is a multicellular organism that is used as a kind of a very nice model to study aging. And I'll tell you why, what is the advantage of it, and what we found. So the first project, most of the work was done by the graduate students at UC Berkeley and Matt Kukurugia. And this was also a collaboration with Saharon Rosette's lab at Tel Aviv University, who is a statistician who helped us with some kind of math aspects of this project. And so before I tell you a little bit about what this Warburg effect is and what is the puzzle, here is some introduction about how our cells, and actually most living cells, produce energy. So in biology, energy comes in the form of this molecules called ATP. So they have energy in a sense that some protein or some enzyme can take this molecule of ATP, hydrolyze it using water, and then energy is released. And this energy can be coupled to just about anything, like, for example, our muscles moving, or us running super fast, or various cells moving inside of our body, or neurons firing and transmitting information. All of this uses ATP as energy. And there are basically two ways that most living organisms use to produce ATP. One that I will kind of call glycolysis. Other names for it is, for example, fermentation. And this is where cells would take glucose, there's a whole bunch of reactions, and they convert it to, so human cells convert it to lactic acid, the yeast cells would convert it to ethanol, and this is the process that leads to ethanol in beer and wine and other drinks. And the second process is respiration, where glucose is also used, but other fuels can also be used with oxygen, and then you produce many more ATP molecules. And kind of key difference here, you get only 2 ATP molecules here per glucose, and you get 20 molecules of ATP here per glucose. So using oxygen and respiration is much more efficient. You can get much more energy. And so historically, people sort of think that this process is only used when oxygen is essentially not available. And then it makes sense, you either make no ATP or a little bit of ATP. Of course, it makes sense to make a little bit. And so, and what Otto Warburg observed more than 100 years ago at this point is that in the presence of oxygen, tumors in rats, for some reason, prefer to use that less efficient way to make ATP, where even though oxygen is available, they for some reason choose to make 2 molecules of energy of ATP instead of 20. And over the years, this has been validated in all kinds of systems. Today, this effect is used to detect tumors in humans in the clinic, where you have this name of FDG. This is basically a radioactive analog of glucose, and PET stands for positron emission tomography. And this technique uses this fact that tumors consume so much glucose to drive this Warburg effect, that they accumulate this radioactively labeled glucose preferentially compared to all the other tissues. And if you use this special scanner, you can actually see it. And then over the last few decades, people have realized that actually it's not really specific to cancer cells only, but in general, kind of bacteria and yeast cells that grow very, very quickly, they prefer to use this glycolysis that seemingly produces less energy. And so the puzzle of this Warburg effect, and so Warburg was a famous scientist in metabolism fields. He got a Nobel Prize sometime in the 1930s for discovering essentially the enzyme that consumes oxygen in our cells. And he made many other contributions to kind of biology and metabolism. And so the puzzle of this Warburg effect that have confused people for a century at this point is, what could possibly be the reason that lets cells choose this process where they can only make 2 ATPs per molecule of glucose, while oxygen is available, instead of using this process that gives you 10 times more ATP. And it just kind of, simplistically, it makes no sense. And it almost seems like evolution made a mistake. And of course, biologists kind of dogmatically believe that evolution doesn't make mistakes. It just, we don't understand what it's trying to optimize. And so what we did in our lab is we tested this kind of a very simple hypothesis, basically suggesting that perhaps the advantage of why rapidly growing cells prefer to use this process is that somehow it is faster. So even though you produce only 2 molecules of ATP per glucose, somehow you're able to do it so fast that you overcome this deficits in efficiency and you're still faster. So that's the idea. And the key question here is fast in which kind of units? And this is where I want to introduce this concept that in our cells are basically a bag of proteins, and the proteins are stuffed inside of our cells to the absolute possible maximum. So there is no space left for any more proteins to be added. So essentially, if our cells want to increase the abundance of some proteins, this has to come at the expense of some other proteins. So it's kind of a zero-sum game. So like, I don't know, imagine you have, I don't know, a plot of land and the zoning rules suggest that you can only build 2 floors. You only can build a house that can be so big. And so cells, the same way. There is no more space inside. And using this idea, what we came up with is that perhaps this glycolysis, let me actually go back here. So perhaps the enzymes that allow these reactions to go through are, very, very small. And you can stuff many, many, many copies of this pathway inside of the cell. And even though you get less ATP per molecule of glucose, because you have many more copies of this pathway inside, you might be able to produce energy faster. So that's the basic idea that we wanted to test. And the way we do everything in the lab, and this is a slide where probably this is too much information, is we use mathematical models to make predictions. So we formulate some hypothesis, then we convert it into equations that can make very specific predictions. Like this is gonna happen, then that is gonna happen, and then we do experiments to test those predictions. If all of those predictions come true, that means whatever the assumptions that we put in the model is also probably true. And so here, our model is pretty simple, where the model basically calculates what would be the rate of that glycolysis pathway and respiration pathway that would allow cells to produce ATP at a maximal possible rate. So mathematically, it looks like this. So it's essentially what's called an optimization problem, where you try to find the maximal value of this function, which is basically this rate of glycolysis plus rate of respiration, and you have to satisfy the constraints. So the constraints here, this constraint basically says that the amount of glycolysis and respiration has to fit inside of the cell. It cannot be more than 100% of protein inside of the cell. And the second constraint says that the rate of glucose consumption by this pathway and that pathway combined is also finite. And we can analyze this model and find solutions to it. And what the solutions suggest is that the Warburg effect would naturally emerge from this if these 2 conditions are true. So one, respiration has to produce more ATP per molecule of glucose, which is something that is well known, and undergraduate students learn it in the introductory biochemistry classes. It's kind of well established. And the second condition means glycolysis has to produce ATP faster. So this is not known, and this is what we went and tested. And the way we tested it is we basically measured it in cells. And so E. coli is a bacteria, as cerevisiae is yeast, and mammals, these are typically, we use human cells. And so the first three graphs show you the yield of ATP per molecule of glucose by this glycolysis pathway and the respiration pathway. And this is where you can see it's 10 times more. And the second row shows you the rate. If you take 1 milligram of one pathway and 1 milligram of the other pathway, how fast will they be able to produce ATP? Yes, question, Joe. I think you are still muted.
Thank you for letting me ask this question. I'm probably getting ahead of you, but if you want to maximize ATP yield by some combination of glycolysis and respiration, cancer cells can figure this out too. So as you try to increase the ATP production, how do you make that effective for the cells you want to be more productive and not for the cancer cells?
I mean, yeah, in terms of, I think what you are saying is in terms of kind of treatments and for cancers, if you want to use this phenomenon to somehow come up with treatments for cancer, then you are completely right. That's we have to somehow find ways to selectively affect cancer cells. And so here what's this project is mostly about is trying to understand kind of the basics of what is the point of it? Why would cancer cells even do it? And if we understand what is the point of it, perhaps this will give us an idea of how to take advantage of it to target cancer cells.
Thank you.
Does that sort of answer your question?
Yeah, it answers it in a kind of a converse way. I understand you find some ways to discourage the cancer cells from being efficient producers of ATP. I take it cancer cells need ATP to grow.
Cancer cells definitely need, yeah, all cells need ATP to grow. And
so in trying to maximize it in the good cells, how do you discourage the cancer cells from picking up on this too?
Yeah, no, I mean, so currently, so I'm not going to talk about it, but we have other projects where we try to use this phenomenon to target cells, and there we do it in a kind of in a very different way by basically taking advantage of the fact that cancer cells, let me see. So because cancer cells do a lot of this, they have a lot of enzymes that are responsible for these reactions. And we found in other projects various tricks about how we can kind of break the regulation of these enzymes to make them kind of futile energy wasters. And the hope is that because cancer cells have a lot of this, maybe they will be targeted selectively, but this is work in progress, very much has to be still figured out. But this project, we don't do any interventions to cancer cells. And we are just trying to understand, what would make cancer cells and other rapidly growing cells to exhibit this phenomenon that seemingly doesn't make sense.
Thank you.
And so what we see from our measurements is basically that the predictions of our mathematical model, the conditions where our model predicts the Warburg effect should occur are very much true. So the first row says that the yield is higher for respiration. Second row says that ATP production rate is higher for glycolysis. And the second row is sort of the key contribution of our study together with the model. And kind of a simple way to imagine what's happening is, so these are the pathways, the two pathways drawn to scale. So imagine this is a cell, and each circle is a protein. It doesn't matter what those proteins are. But the red circles are the proteins that are part of this glycolysis pathway, the one that produces less ATP per glucose. And the blue circles are parts of the respiration pathway. And this is the amount of enzymes that is required to catalyze, to consume one equivalent of glucose. And you can see that this respiration is just giant. It is approximately 30 times bigger than the glycolysis pathway. And kind of the interpretation of this is that, cells basically have a choice. They either invest lots of resources into making this giant pathway, and then they can be very efficient at producing energy, or they can spend less resources making this tiny pathway, but because it's tiny, they can make more copies of it, and this way they can produce energy faster. So kind of perhaps like an analogy could be, you can build a nuclear power plant, but nuclear power plant costs billions of dollars. And I mean, these days with all of the regulations, it costs tens of billions of dollars. And so if you want your iPhone to work, you're not going to build a nuclear power plant to power it. You're going to insert a small battery that's going to cost nothing, but it can produce less energy, but you can use more batteries. And so that's basically the choice that cells can make. And so then finally, this is the final slides about this project. The way we kind of typically test or try to prove whether our model and hypothesis is correct is we have a model that can make predictions, and the various parameters of these models are measured from one set of experiments, but then model makes predictions about completely different sets of experiments. And this is what we did here, where we have our model that can make predictions based on some purified protein experiments. And then we compare the predictions to live cell experiments. And so this is, each dot is basically data that's we either generated ourselves or we found in the literature for this bacteria, yeast, and mammalian cells. And the lines and the ribbons around the lines are the predictions of the model. And you can see that the model can reasonably accurately predict essentially all of the experiments that have been done previously investigating this process. And kind of we interpret this types of observations as a very strong evidence that whatever the assumptions that we've put into the model is actually how things work. So this is one example of the project that we work on. And basically the summary is this mysterious phenomenon is happening because it allows cells to produce ATP faster by sacrificing the efficiency of the process. Any questions about any aspects of this first project / question?
Roger has a question. Would you unmute yourself, Roger?
So can you hear me now?
Yep.
Yeah, okay. So I was struck in your this slide that you have up, the mammals are very similar to yeast, but very dissimilar to the E. coli. And I wonder, does that indicate a turning point in evolution when some very primitive creature hit upon this pathway and then it was inherited through both lines? Is that a good interpretation of that?
Yeah, I mean, that's an interesting question. So the differences, I mean, on this type of plots, they look quite dramatic, but it's actually just a few quantitative changes in essentially the same processes lead to this difference.
Mm-hmm.
And so for some reason, evolution, so let me go back to so for example, so this is for humans. So in bacteria, evolution, there was some kind of evolutionary pressure that made many of these blue enzymes way smaller. And they still, as far as we can tell, do exactly the same thing, but they are much smaller. And so because of that, even this blue pathway can actually go faster. And so because of that, bacteria, basically the red thing, never goes anywhere. So in bacteria, this respiration pathway is often preferred. And this is why that happened exactly, I think we don't know the answer to. but somehow evolution decided it was much more important for bacteria to grow super fast compared to human cells, and they optimized that quite a lot. And so from other experiments, we know that you can put bacteria in some conditions, and within 100 generations, they can accumulate mutations that would allow them to grow faster in that specific conditions. So human cells just don't do that. I guess they're not evolving as fast. Perhaps they're less optimal.
So maybe that's because the generation time is so long with humans.
Yeah, so maybe another way to think about it is basically E. coli is much more evolved because they went through way more generations since our last common ancestor compared to humans. And yeah, another way to look at it is if you look at the values on the Y scale, like you can see that bacteria, or maybe even, let's say that this plot. So this is ATP production rate per milligram of pathway. So you can see the human respiration is 0.5 in these units, but bacterial respiration is 10. So it's 20 times faster, even though just kind of reactions are exactly the same. There is no difference in the reactions. It's just the proteins became more compact and faster at performing the same function. And it's, yeah.
Can I ask another question?
Please.
So in my backyard, we have a compost heap. And the word about compost is that you should turn it frequently because you want to get the aerobic bacteria in there and not the anaerobic. So that tells me that there's some important difference in terms of I think it's basically related to the speed at which the compost breaks down. If you just leave a stack of leaves or something and just leave them there, they get wet, they get soggy. It takes a really long time for them to break down. But if you turn them frequently, they break down a lot more quickly and you can use it in your garden. So does that have anything to do with this these pathways? Because the anaerobes wouldn't be using that blue pathway, isn't that right?
Yeah. I mean, I don't know too much about compost, but just kind of naively, this 20 versus 2, I mean, it's still there. So basically, if you're an organism that uses oxygen, you can extract much more energy from the same one leaf, let's say, and you can grow to become a much bigger population, so you can eat way more leaves much faster. Just because, another way to say it, this phenomenon is very much for the situations when the conditions are just perfect. You are just swimming in like unlimited amounts of fuel and nutrients, and you wanna, in that condition, you wanna go as fast as possible. But in nature, that's never a thing. You are always limited by resources, and that's why, all of the organisms essentially have this pathway in addition to this pathway. In fact, some organisms are missing this pathway because it's just not useful for them in their environment. They never experience conditions where the nutrients are in such a large excess that it makes sense. One analogy different from compost is, let's say, so human muscle. So we have two types of muscle cells, simplistically. One type uses this process, and this is if we want to walk for a long distance or like jog lightly for long distances. Our muscles essentially use oxygen and they make lots of energy. But if we want to do very short and like single-digit second or tenths of seconds burst of exercise, we have a different type of muscle called fast twitch muscle that essentially only uses this. It doesn't use oxygen. And so if you want to do short bursts of lots of energy production, you go with this pathway. But if you just want to get further, this is the way.
Okay. Okay, I got another question.
Yeah, please.
Well, you don't have any plants in there.
No
And
plants.
so, I mean, plants are a pretty important part of the biosphere. And do they must be making ATP, I would imagine. This ATP thing goes back evolutionarily, goes back before the evolution of plants, doesn't it? So
Yeah, ATP is universal. Yes.
it's universal. So it's right back there with DNA, and you know, it's a very basic, or I guess RNA is even more ancient than DNA. So what about plants? Are they using, which pathway do they use?
So I mean, plants, being plants, they do photosynthesis. So they make ATP from light, from sunlight.
Mm-hmm.
So actually, they do the reverse. They do the reverse of this pathway.
Oh.
Basically, exactly the reverse. So they take, I mean, essentially water and CO2, and then sunlight, and then they produce oxygen and tons of it. And because there is so much energy in the sunlight, they not only go backwards, but also produce tons of ATP in the process, and also produce the glucose and everything. So it's a photosynthesis is a, this is only possible because plants, and before plants, cyanobacteria produced all of this oxygen. And so all of the oxygen that we breathe, it didn't exist without life. It's all thanks to life that oxygen was produced. And so this is, in some sense, is something that's had to have evolved way later, like consuming biological products and consuming oxygen, which is also a biological product. Yeah, it's kind of fascinating, I don't know, history.
Yeah.
Now that's now very popular with powerful telescopes to, we have technology to look for, life on other planets. And the best way in which we can detect it is by looking at, for example, oxygen. If we go and with a telescope, when the planet passes in front of the sun, we can analyze its atmosphere of the planet. And if we see that there is a planet with 20% oxygen in it, or more oxygen, there is basically no chemical process that we know of that would do this except for life. So this is how we would know that planet has life. Whether that life, has neurons, brains, and is sitting there and looking in telescopes, we won't know. But we will know that something has produced that oxygen.
Okay.
Yeah. Which that's obviously an epic tangent compared to what I was talking about.
Okay, anybody else before I ask another question? So I volunteer at the Academy of Sciences in San Francisco, and I work explaining the coral reef organisms to the visitors. And there are some very interesting organisms there, which are animals, like anemones, for example, or corals, or a lot of these they're animals. They're stationary. They don't go anywhere, except maybe as a larval stage they move around. But once they're developed into adults, they're stationary. And one of the ways that they get food is through photosynthesis, which is not supposed to be happening with animals, right? But what they have is they have these commensal bacteria are in their algae. They have algae in their cells. And so these cells, they've actually, as I understand it, they've actually incorporated them into their cells, just like our cells have these organelles that probably came from some other life form originally. But what I'm wondering about is the production of ATP in an organism like that, which is a hybrid, I guess you could say, or a combination. So would they be using both pathways, or is there any opposition there, or do they is it just cumulative what they are able to produce?
So I mean, plants have both. So they have the chloroplast, which is the thing that uses sunlight to make ATP, but they also have mitochondria, which is the thing that uses then oxygen and burns glucose to make ATP. And like the simplistic explanation is, sometimes it's dark and you have to stay alive while it's dark. And so they have light and dark cycles where they switch their metabolism from making ATP from sunlight, and then no sunlight, they start making ATP by burning resources. And so there's lots of, exactly what happens in corals, I don't know, but there are lots of examples like that, even in plants.
So they can do both. They profit from both.
There's
no conflict there.
There is a, like, you don't want them to be working necessarily at the same time. They there is lots of kind of chemical details that you want them to be working separately. Otherwise, it'll be just kind of a futile cycle. Probably some things will go wrong, but I don't know too much about how those two processes talk to one another. Probably if they both work at the same time, something bad might happen.
So they have some kind of a switch or something that they switch from one form to the other.
Yep.
Yeah. Okay.
Yeah, even in humans, we have circadian rhythm where the type of proteins that our cells have changes throughout the day, depending on sunlight that we see through our eyes. So, and plants surely have even more things like that.
Okay. All right. Thank you.
Okay. So 341. So let me tell you about the other thing, which is completely different. So the question here is, so there is this process called caloric restriction, and we want to know how it works. So people who did this work, so most of it was done by Bowen, who was a graduate student at UC Berkeley, and it was a collaboration with Aaron Streets, who's a professor at UC Berkeley, and Bryce is a postdoctoral fellow in his lab. So they are at the bioengineering department. And so calorie restriction is this another century-old puzzle where people have figured out first in rats about, I think, slightly more than 100 years ago, that if you basically give rats less food, they live longer. And so here, the way this is done is you have rats that have foods always available to them in their cage. And so they basically, since they don't know any better, they don't see commercials on TV saying don't eat so much, they just eat continuously, and they're in red. So this is their lifespan. So rats live for about 2 years. So this is their lifespan, the red ones. And then the green rats are the rats where the investigators measure exactly how much food did the red rats ate, and then give them exactly 40% less. And the green ones, instead of living for 2 years, they live for 3 years, which is a major difference in lifespan. And so over the last 100 years, basically every organism where people have tried this in, this works. The effect size could be different. In some organisms, it can be bigger. In some organisms, it could be smaller, but always it is better to eat slightly less food compared to what organism would eat if food was available continuously. And for humans, there is this experiment, of course, is very difficult to practically do and probably will never be done. But there is tons of evidence in humans that the same thing happens, where there is all of this association of body mass index to many chronic diseases. Now with this new generation of appetite suppressing drugs like Ozempic and a bunch of others, now we have clinical trials where we can see what happens if you eat less for a whole bunch of chronic diseases, like you have improvements in symptoms. And so the question is, how does this work? And since this works for essentially every organism where this has been tested, in biology, typically people try to choose the simplest possible organism and figure out how this works over there. And if the same process is conserved everywhere, then hopefully the lessons that we learn from simple organisms will be transferable to humans. And so what we chose to use is this organism called C. elegans, which is very, very popular model organism. Many discoveries in biology about how multicellular organisms work have been done in C. elegans. And so it's a tiny worm. So this is its life cycle. So they live for about 2 to 3 weeks. And so the worm, basically, each worm lays, so there can be either males or hermaphrodites. Hermaphrodite means they self-fertilize. So you can have one worm can produce 300-400 progeny without anybody else. So they can basically clone themselves, which is very convenient from an experimental point of view. You don't have to mate them, do sexual reproduction, which takes a lot of time and effort. And they pretty quickly from the embryo, essentially in 2 to 3 days, they become an adult that is capable of reproduction. So again, from a convenience and science point of view, we can grow giant populations of the C. elegans and do all kinds of experiments with them. And so like all the other animals, C. elegans, if you give them less food, they live. So here, instead of 2 weeks, they live, so here, if you use optimal conditions, you essentially double their lifespan if you give them less food. And here food means, so they eat bacteria, so we basically give them less bacteria. So these are C. elegans. And we have various fancy machines so C. elegans, their genome is sequenced. We have techniques to essentially manipulate any genome we want in any way we want. In 2 to 3 weeks, we can have an organism that is altered in whatever way we want. So very convenient, lots of tools to study them. We have large machines like this, where there are lots of scanners inside of this incubator. And what the scanners do is they measure the lifespan of these worms. So here, this is a plate. The large dots over here, this is a lawn of bacteria. And the tiny things, let me see, can I click on it again? So the tiny wiggly things are the worms. So this is 3 weeks in 10 seconds. So you can see that, so this is the number that you might or might not see is days. So you can see that they wiggle super fast. After about 2 weeks, they stop wiggling. And when they stop wiggling and never wiggle again, we assume that they died. And we have automated software that processes all of this, and we can draw this lifespan curves with this machine. And with the current setup that we have, this is actually outdated now. We have 2 machines like that, so we can measure lifespan of about 10,000 of these worms. And then we basically do various experiments, measure lifespan, see how it's affected, and try to figure out which genes, which processes leads to this doubling of lifespan with caloric restriction. And so one of the really interesting experiments that we did is in collaboration with the Streets Lab that has, so they use this very fancy microscopy called stimulated Raman scattering. So essentially, it kind of works like a microwave oven. So in a microwave, you use those microwave wavelengths to basically heat up the hydrogen, oxygen bonds and water. So you heat up water, it starts wiggling, moving faster, and you heat up your food. But here, instead of doing it with water, which is over here, this microscope can detect the CH2 bonds or CH3 bonds. And you don't need to label your organism in any way. You just look at those worms. And another beauty, another of the many convenient things about worms is they're transparent. So we can, with microscopy, we can see any parts of the worm. And so basically with this microscopy, essentially what we can do is we can take pictures of where the proteins and where the lipids are inside of the animal. And so we use this microscope to take pictures like this. So here blue is protein. So we don't know what protein. All we can say is it's protein. And the yellow is lipids. And so maybe let me highlight a few interesting. So this is C. elegans. This is its head. So this is a zoom in of the head. It has this thing called pharynx. So essentially, so this is the mouth. It opens, it like swallows a bunch of bacteria. This thing is some kind of like a grinding machine that it basically takes the bacteria and grinds them up before swallowing them. And this is, so C. elegans, so they have about 1,000 somatic cells and about as much of the germline. So it's essentially, I don't know, like a intestine attached to the germline. So very, I don't know, very efficient. And so there, after they swallow things, let me just go back to this image. So here, this is the head. This whole thing, this is the germline. So it starts with like amorphous blob of nuclei, and then it becomes cells, and then becomes, this is an oocyte. And the oocyte then matures, and this is a fertilized embryo. And they have one germline, so-called gonadal arm on one side, and there is a symmetric arm on the other side, which we cannot see because it's underneath the intestine. And this is the intestine. So here the bacteria are swallowed, they go into the intestine. There is intestine underneath this thing that we cannot see, which goes, and then it comes out over here. And the line over here is the lumen of the intestine, and the surrounding things are basically intestinal cells. And in this worm's intestines are basically like intestines plus kidneys plus liver plus adipose tissue combined in one thing. And that's why you can see so many lipid droplets inside. So these are worms. So here is the zoom in, for example, on the embryo. You can kind of see, so here this is a nuclei of one of the cells. You can see it has a bunch of cells. And this embryo in 3 days will turn into adults that is this big. And one of the actually cool applications that people use C. elegans for is to study developments, because it's so, C. elegans are a type of organisms that produces exactly the same number of cells in exactly the same way every single time. So they have something like 1,076 cells, and people have figured out where does each single cell comes from, and it's a very convenient model to try to understand, like, how is this even possible? How did biology come up with ways to convert this blob into this organism in like 2 to 3 days, which is amazing, and we only have very little idea of how this works. But that's not what I study. So anyway, so this is intestine again. So this is the lumen, where the food is basically getting digested, and then it's stored in those various vesicles. Okay, and so then what we did is we took thousands and thousands of pictures of these worms. This is the young worm, so day one. Day one meaning day one after it became reproductively active. And this is the older worm, so day 6. So remember that with lots of food, they live for like a little bit more than 2 weeks. So this is old, but not too old. And what's kind of the most striking observation was that there is this kind of amorphous protein blob that accumulates. And when we look carefully, we see that it is extracellular, so it's outside of the cells. So it's basically some kind of protein that is secreted in just giant amounts outside of cells, and you can see it accumulates just everywhere. And I guess in general also you can see that here there is some structure and it looks kind of beautiful, symmetric, and here it's only 5 days later, just everything kind of looks like a mess. It's hard to say what is what. There is this beautiful arm is not visible anymore, and it's just kind of things look like they're falling apart. So this is what we saw. And then we did a whole bunch of various experiments and essentially figured out what this protein actually is that accumulates. And it turns out that this is a protein called, a class of proteins called vitellogenins. And vitellogenins are basically, so in worms, so the intestine, accumulates nutrients, and then it packages them in these proteins called vitellogenins that are then secreted from the intestine. So over here in the intestine, they're packaged, they're secreted outside, and then they go to the oocytes, and they're taken up by the oocytes. And so this is essentially, this is yolk protein that is produced, because in C. elegans, the embryo, once it leaves the parents, it's actually not like a moving worm yet. So it continues developing outside of the parent's body. So it's worms essentially that lay eggs. And every egg-laying organism, from these worms to spiders to chickens, have these proteins called vitellogenins, which are almost identical in sequence. So the chicken yolk in the eggs has basically the same protein. And so you're conserved in all of the egg-laying animals, and it turns out actually it's even more interesting. So this is exactly the same protein that in humans forms the LDL particles. So it's the same protein that originally evolution used it for this purpose. And for worms, it kind of makes sense because the eggs is the majority of the nutrients of the whole organism actually goes into laying eggs, because each organism can lay about 300 eggs in their lifespan. So they lay, their body weight in eggs every day. So it's just kind of crazy amounts. And so in terms of where do all the nutrients go in the worms, all of the nutrients go into making new eggs. And so of course in humans, it has changed where LDL is used mostly to feed the rest of our tissues. But the protein is this vitellogenin protein is homologous to LDL. LDL protein has receptors on all human cells. That receptor is homologous to the receptor that worms use to take this up. So for all intents and purposes, it's kind of a similar protein. And there are, of course, some differences, because in this vitellogenin proteins, actually the protein itself is the cargo. The protein itself is injected and is destroyed for amino acids. And in LDL, actually the lipids inside of the particle are the cargo. But this apparently is a small adaptation. Okay, and so then basically the summary of what we found is, we found this protein accumulates in old animals. We found that this is the same protein that is homologous to LDL in humans. In experiments that I didn't show you, we found that if you actually calorically restrict animals, the amount of this vitellogenin protein goes down. And so now the obvious hypothesis is maybe this is how lifespan is extended. And for this, we did an experiment where basically, so think of this, so if caloric restriction extends lifespan by decreasing these proteins, then what we should be able to observe is by just decreasing this protein, we should see lifespan extending without doing any caloric restriction. And so this is what we did. So here, fed worms live this long, calorically restricted worms live this long. If we delete, in worms, you can actually remove all of those vitellogenin proteins. And sort of surprisingly, they produce as many eggs as before. And the only phenotype is if the eggs are put in a condition where there is no food, then they can only survive for 1 day. But in the present, if the parent was able to produce vitellogenins, then the eggs can crawl around for 3 weeks. Not the eggs, the organism that hatches from the egg. So it's kind of a, let me say it again. Basically, if a parent is able to do this vitellogenesis, then the eggs that are laid in the complete absence of food, they hatch, and the little worm, it cannot grow because there is no food, but it can crawl around for 3 weeks, presumably looking for another rotten apple to eat with bacteria. But if the parent is not able to do vitellogenesis. This egg can still hatch, but the little worm can only crawl around for like a day before it dies from starvation. So it provides basically energy reserve for the animals to be able to crawl around, which evolutionarily, when they live in the wild, is probably a huge advantage. So when we delete all of this vitellogenins, the worms also live almost as long as calorically restricted worms. But if we do both together, you can see that's either caloric restriction or caloric restriction without any of those vitellogenins. Basically, the lifespan is the same. And this is the type of experiment that people in the fields often do to basically provide evidence to the idea that this is a linear pathway where this thing downregulates this. And because this is downregulated, lifespan is increased. And we also have some other experiments basically showing the same thing. And so, okay, what time is it? 4. So the summary is, so what we found is that caloric restriction in this elegans appears to be inhibiting and extending lifespan by inhibiting this vitellogenesis. And by analogy with sort of humans and other organisms, it seems that perhaps it's an evolutionarily conserved mode of aging in response to consuming too many calories, where you overproduce this nutrient carrier proteins that maybe are beneficial for producing lots of progeny super fast, but it shortens the lifespan of the parent. And there are, of course, many unanswered questions where with this C. elegans, because they're so simple, we kind of have a chance to study how does excessive production of the secreted protein affects lifespan, because there are many diseases in humans that have a similar mechanism of there is a secreted protein that is secreted too much for one reason or another. So LDL cholesterol is one, so it's secreted in response to diets, but also in response to aging. Similarly, there is AA beta protein and Alzheimer's disease, where this is a secreted protein that's, for reasons that are not well understood, sometimes is secreted much more, causes pathology. Similarly, there are various diseases called amyloidosis, where different organs can be affected by proteins that are secreted in much larger amounts than they should be. And with this simple system, the hope, because we can do this microscopy, and this is endogenous protein, we can find some kind of better molecular understanding of exactly what happens and why this is toxic to the organism. And hopefully some of those findings will also be applicable to humans. And so this is all I wanted to tell you about. So this is our lab. That's most of them do various other projects that I didn't talk about. And this is who funds our research. You know, almost all of our research is from NIH. So, and thank you for your attention. Happy to answer any more questions.
Thank you, Denis. This was really fascinating. Can you tell me first, when was C. elegans really discovered? Have we known about it a long time?
Discovered, or when did they separate from humans?
When in the lab you discovered it was such an interesting tool for your research, perhaps.
So C. elegans, I mean, it's a workhorse model organ, and people have been using it for decades. So
yeah.
probably since like, actually, I don't know, let's say since the 70s. So they're not as old as fruit flies, for example. Fruit flies, people have been doing genetic experiments on fruit flies for more than 100 years. So C. elegans are more recent.
This seems so much, I mean, this to the layperson, such as myself, this was fascinating because, first of all, you could show me something that was a size I could grasp. And also you have such detail in your, with the spectrum.
Microscopy.
Yes, yes.
Yeah.
And also you have already, you know, such a thorough understanding of the process leading to us being told to watch out for LDL. So the oxygen related that you talked about at the beginning, the oxygen intake, if we increase our oxygen intake along with watching generally things like LDL, that is theoretically then conducive to aging, unless we have bad genes or something else, right?
Conducive to aging, meaning
And it would be
more faster.
if we limit our LDL and we take in lots, reasonable amounts of oxygen, keep moving, etc. In theory, in a perfect situation, we would age more. But obviously, there are so many factors that influence aging.
Like age slower, you mean? So I mean, like the LDL.
Yeah, we would age more slowly. Yes,
Yeah,
thank you.
yeah.
Yeah.
So I mean, LDL is a, if metabolism, nutrition, and just medicine in general have figured anything out over the last 50 years, it's the LDL is it. I mean, the fact that high levels of LDL cholesterol cause the majority of heart attacks, which is the #1 cause of death in the world by a lot. And this is the cause. And LDL levels can be lowered by an order of magnitude in an average person in any developed country with no side effects that people know of. So in a sense, what we know now, as long as kind of, I don't know, either modifications and diets, which practically doesn't seem to work. So in reality, it means drugs. If drugs are used to lower LDL levels are used more aggressively, and people are more aware that this is possible, heart disease can probably be decisively reduced by many, many, many fold, which is there's no other disease that we have anything like that. It's almost like a paradoxical situation. And so, but with oxygen is very complicated. So oxygen is it's difficult for us. Let's say if we try to breathe like higher concentration of oxygen, so that's bad. So it's in a sense, by higher oxygen, I think what you meant is kind of more exercise, more kind of energy. So and that's that's also can affect LDL, and it has some other beneficial effects that I think we're not fully understanding yet. But it is 100% beneficial. It's just the amounts is the only question. And so with exercise, I mean, the other interesting thing about exercise is it suppresses appetite, which is kind of almost doesn't make any sense. Like you went, let's say, for a long walk or for a run, and it might sometimes feel that you are more hungry, but the research very clearly shows that it actually suppresses appetites in people. So in some sense, by exercising, you also eat less, which is very, very confusing. I don't know why evolution did it that way. I don't understand the logic.
So under the umbrella of some of your work, which is nutritional science and toxicology, it seems like you have a real blend between molecular cell and computational, but also under nutritional science and toxicology, where are you going within that umbrella?
I mean, it's more, I study like a molecular nutrition is more or less what I study. And the department, I mean, historically it has a name, nutritional sciences and toxicology, but I think we are in the very long bureaucratic process of dropping the toxicology parts because people actually don't study it in our departments anymore. So it's going to be, I think, renamed to molecular, what? Now I forgot. Molecular metabolism and nutrition. But what I study is very much understanding molecules and how the processes work in a very kind of basic science way. And I don't even do any experiments in mice. Many of my colleagues in the departments do experiments with mice, but I just do the simplest organism imaginable. And we do lots of actually computational work that I didn't really talk about, where we basically try to build bigger and bigger models to, I guess the way I think about it, the future of biology, where one day, some decades later, we will figure out how everything works in our body, and we'll have a similar understanding to it, how we understand exactly how our iPhones work. That's the way it will look like.
Thank you.
Large-scale computational models. And now metabolism is actually one of the areas where this will probably happen first, just because people have been making quantitative measurements for much longer. And it's clear how to make that next step. Now that computers have become super fast, it's very easy to generate lots of data. That's kind of the direction where we're mostly going.
Thank you. Thank you. Other people have questions.
Yeah.
Thank
yeah.
you.
Question?
Joe has his hand up next.
I do. Some number of years ago, there was an article in the Chronicle about a club somewhere in the East Bay, Berkeley, where people were practicing that. In other words, severe decreases in calorie intake.
Yeah. Yeah.
Have you seen any data on that and how that's going?
I mean, let me say something completely different first. So I think the calorie restriction, I think the term is very unfortunate. Kind of the term is historic. It is called calorie restriction. I mean, really, if I were to name it today, it would be like not ridiculous overeating. Because we are comparing, all of the experiments are done compared to an organism that eats continuously. And let's say so everyone here is calorie restricting, whether you know it or not, because we know that if we eat breakfast, lunch, and dinner like Big Mac, like this is gonna be very bad on many levels. And so our doctors tell us to watch our weights. We know that, I don't know, we need to eat less fast food, etc., etc., etc. So all people are already calorie restricting to one way or another. And so it's an interesting question. What is the optimal level? Like how much do you need to eat to achieve optimal health benefits? And unfortunately, it's very much unclear, because it's very, the way to do it would be to take people who eat different amounts of food and do giant experiments like that. And it's not practical. And maybe with the Ozempic drugs, we'll have some better sense of how to do it. But basically, that it's not at all clear if you take people with a normal BMI and somehow they convince themselves to eat less and lose more weight, what this will do is not clear, because there are eventually negative effects where you get more prone to infections and things like that. So it's not really, in terms of the studies in humans, so they do, I think there is one large-scale clinical trial, it's called Caloric. That's some clever acronym of what calorie stands for, but they somehow, because it's very difficult to tell people to eat less and then to check whether they actually did it or not, whether people try it or not. People do large-scale trials to try to modify diets. It just doesn't work. Everybody can lose weight for 6 months, and then if you check 2 years later, on average, everything is back to where it started. It's just not something that people have figured out how to do. That's why this new generation of drugs that's, I don't know, are imperfect in various ways, but they are the first way that's people figured out how to durably lose weight. And
Thank you.
This is the first time that we're able to actually see all of this data. That's all of this findings that we have from model organisms, that we can exactly know how to give them less food because we're in full control. And we see all of the same effects essentially observed in all of the Ozempic trials, which suggests that many of the things that we found in simpler organisms is transferable to humans. But practically, what to do and how much to eat, unfortunately, who knows? And it's probably dependent on the person, depends on what you eat, etc. And I guess while we're talking about it, the very good studies that people do on humans are of the type where you take a relatively small number of volunteers, let's say 50 people, and then they stay in the clinic for, let's say, 3 weeks, which is, immediately you can tell that this is very hard, like to convince dozens of people to sit in some room for 3 weeks. It's very hard. But because But if you can do it, you can do very careful studies where they provide all of the meals and they can modify the meals in various ways, and they can do lots of measurements, and they can see basically what about a meal makes a person eat more or less. And these are the type of studies where people have very clearly shown that eating so-called ultra-processed foods, so basically think of anything that you look at it and it does not look like a plant or a piece of meat at all. That's ultra-processed foods. And if people are fed ultra-processed foods versus things that still look like food, people who eat ultra-processed foods consume on average 300 calories more per day, which is a huge effect. And so in some sense, I don't know, perhaps the way out of this is some kind of change in the food industry to convince companies to produce less ultra-processed foods. But I'm sure Coca-Cola will be very much against this, and they probably can lobby Congress better than just about anybody. So hard to do it in practice. So I didn't answer your question because
No,
there's no answer.
that's fine. I think we have time for one more question.
Joan is waiting. Go ahead, Joan.
It's going to be a short one. First of all, thank you so much, Dennis. This is really, really interesting. In terms of diet, this Dr. Brednison, in terms of working with Alzheimer's patients, talks about fasting 12 hours and eating 12 hours. Do you have any experience with whether that's an effective way of restricting calories?
I mean, it's a very popular thing, but I don't know of any very large scale trials that demonstrates that this works. And it's probably basically anything you can do to convince yourself to eat less is great. Whether it's skipping breakfasts or only eating once a day, or if you eat basically only salads, like physically it's impossible to chew so fast to consume as many calories as compared to eating some other things. Or there's, I don't know, exercising a lot, but it depends. Different people are different. Different people like different things. But in general, whatever you do to convince yourself to, I don't know, not eat too much, probably works.
Thank you.
That's eating during a 12-hour window, then it's going to work. And if that's not, it's then probably not going to work. But no large-scale trials showing that this is highly effective that I know of.
Hey, Roger, you had another question?
Oh, yeah. So, yeah, the question I wanted to know is there anything politically incorrect about your work that threatens your funding?
Who knows? Who knows? I think that's a hard one to answer, because who knows exactly what the current administration wants from science. Yeah, it's hard to say. I mean, what I study seems like very much a no-brainer in terms of investing resources into understanding all of those processes more. But it's a complicated thing. Like I mean, I presume you are referring to many changes that seem to be coming every day from the current administration, proposing reforms to the NIH and things like that.
Yeah.
And it's hard. I mean, the only thing I can say about that is there's discussion about something called indirect costs that is ongoing currently, which is kind of unfortunately misrepresented a little bit. So the way the NIH and federal agencies that sponsor science as they give $1 to the researcher to do like the actual stuff, to like pay salaries of the people who physically do the research, to buy the reagents, to do blah blah blah. And then for every dollar, about 60 cents goes to the university. Because like I sit in a building, somebody has to make sure this building doesn't fall apart, and we have electricity, which are the simple things. But the more complicated things, we have very expensive facilities that maintain equipment that one lab cannot buy. And those facilities are being paid by this indirect costs that NIH provides. And whether this should be this 60 cents for every dollar or 55 or 70, I mean, who knows? But all of that money is carefully accounted for. You can read where every cent goes. And the proposal to cut that from, so for UC Berkeley, it's 61 cents per dollar to 15 cents. If
Wow.
that goes through, then all of the science in US will go bankrupt overnight. There's just no way people can support this because it's basically a cut, a 1/3 cut to research budgets.
Hmm.
Presumably, most of Congress understands this, and this is just a negotiation tactic to do something, but who knows?
Yeah, well, good
In
luck.
terms of specific universities, how the Columbia University, or at least the government, the federal government proposes to take away all of their grants for various reasons. I mean, it's difficult to comment about that, why that is and what is the point of it.
Yeah.
Not clear to me. But yeah, in terms of specific research problems, I think there is, well, the only targeting of specific research problems that I'm aware of is infectious disease, where I think the current administration proposes to decrease spending for infectious disease specifically, which is, I mean, it's difficult to say something about that. Feels like a mistake, because given the recent experience, probably need to spend more money studying infectious diseases to understand better. And I guess the other programs that are being cut, where I have friends doing this, is the US had a program actually started by the Bush Jr. administration that was very effective at controlling AIDS in Africa, where, I mean, it was significant amount of money, several $1 billion, not significant on the scale of federal budgets, but it's significant on the scale of this problem. And they were very effective. They were making a big difference, and the kind of the AIDS and the HIV prevalence in Africa was plummeting as a result of it. And that program was funded by USAID, and that is proposed to be cut. If that happens, I mean, it's not going to affect Americans directly, but it's probably doesn't seem like the best program to cut because of how successful has been demonstrated to be.
Yeah. Well, these are perilous times. Yeah.
Do we have any more questions? If not, you've heard it already from a couple of people, but wonderful presentation today.
Thank you so much, Denis.
Enjoyed it.
You've been very generous.
Thanks
Thank
so
you
much
for
for
your
inviting
time.
me. Yeah, it was great chatting.
Such important work too. So thank you very much. And we'll see you all next month.